So what I'm saying is that we can manage cancer very, very effectively in the majority, if not all, cancer patients by lowering the fermentable fuels, glucose and glutamine, and transitioning the whole body to ketone bodies and fatty acids. Because the liver burns fatty acids and the rest of the cells, the brain, can burn ketone bodies, all organs can burn. Cancer cells cannot burn fatty acids and ketone bodies. They store them as lipid droplets. They're not burning them.
Welcome to the Low Carb Health Doctor podcast. So every so often, I make it a point to delve into the minds of leading experts in metabolic health and science to help us all understand how to effectively reverse hypertension, diabetes, obesity, and with the overarching goal to really fight metabolic syndrome. So, cancer is one of the very serious health problems in the world today. And based on statistics from the World Health Organization, it is among the top 10 most significant causes of death globally. And today we shall be talking about cancer. And we are very much honored to have as our guest Professor Thomas Seyfried. Professor Seyfried is a distinguished American professor of biology, genetics, and biochemistry at Boston College. He holds a master's degree in genetics from the Illinois State University, and he received his Ph.D. in genetics and biochemistry from the University of Illinois, Urbana-Champaign. He further honed his expertise with postdoctoral studies in neurochemistry and genetics at Yale University's Department of Neurology, where he served as assistant professor. Professor Seyfried's illustrious career spans over 200 peer-reviewed publications, and he was focusing mainly on the mechanisms underlying chronic diseases such as cancer, epilepsy, and neurodegenerative disorders. Currently, he serves on the editorial boards of several journals, including Nutrition and Metabolism, the Journal of Lipid Research, Neurochemical Research, and ASN Neuro. And his groundbreaking work with his preeminent book published by Wiley on cancer as a metabolic disease has positioned him at the forefront of cancer research worldwide. So I encourage you all to join us as we explore cancer as a metabolic disease in this very enlightening conversation with Professor Thomas Seyfried. Professor Seyfried, a very warm welcome from the Low Carb Health Doctor platform.
Well, thank you very much, Dr. Don. It's a pleasure to be here and have the opportunity to speak to your following guests.
So first things first, I think everybody would like to know about your early beginnings in cancer research, Professor. So what I am particularly interested to ask you is, how did you eventually come across the concept of cancer as a metabolic dysfunction or a metabolic driven disease? What got you most interested into doing research about it?
Well, yeah, thank you. Well, I did not start at all my scientific career working on cancer or anything to do with cancer. My studies were basically in mammalian genetics and biochemistry. I was looking at first at Illinois State University. I studied on a preeminent geneticist, Dr. Herman Brockman, in neurospora genetics, looking at the effects of mutations on adenine pathways in neurospora. Mutagenic, we were looking at different mutagens causing mutations in this bread mold. Then I went to the University of Illinois and I studied mouse genetics, mainly looking at lipid storage diseases. I became fascinated with the field of gangliosides, which are very complex sialic acid containing glycolipids. I got a postdoc to follow this work with Dr. Robert Yu at Yale University, the late Dr. Robert Yu. And we started looking at these glycolipids in epilepsy. I was in a department where the focus was epilepsy and biochemistry. So in order to stay there, Dr. Yu said, you better do something on epilepsy. So I started looking at various epileptic seizures in mice, mapping genes that might contribute to dysregulated excitation in the brain. And at the same time, I was trying to figure out what these incredibly complicated lipids were doing in the brain. So I was doing developmental biology, biochemistry, genetics, all these kinds of things.
That was in Yale University, right?
Yeah.
Yale.
Well, it started at the University of Illinois, and then I took it on to Yale. I spent 9 years there at Yale, first as a postdoc, then as an assistant professor. And Dr. Yu had looked at some ganglioside biochemistry in tumors, and I thought it would be a good idea to also study gangliosides in tumors, not because of any desire to cure cancer or understand. It was more to use cancer as a tool to understand gangliosides, not the reverse. But we made all these different brain tumors in mice so I could study the ganglioside distribution in the brain tumors of the mouse to see whether or not there might be any similarities with human brain tumors. And I learned a lot doing that. And at the same time, I was mapping genes for epileptic seizures. So we were doing a lot of research. And then I took a position at Boston College in their biology department where I could interact more with students, because mostly at Yale, you're dealing with professional scientists, postdocs, and this kind of thing. Whereas at Boston College, you're really working with undergraduates and graduate students. And then I brought all my scientific technology from Yale University to Boston College, and also the various animal models that I developed at Yale in both epilepsy and brain cancer. So I had funding from NIH to study epilepsy and map genes for epilepsy, and then I got grants to study the role of gangliosides in angiogenesis in tumors, which is the role of angiogenesis, abnormal blood vessels. And around the late 90s, early 2000, angiogenesis was a hot area in cancer. So I had found a ganglioside that would inhibit angiogenesis. So this then gave me a couple of grants, but it was nothing to wonder about the nature of tumors and how they grew and I wasn't thinking about anything about that. And I, like everyone else in the world, thought cancer was a genetic disease because I taught students out of textbooks and things like this, and I didn't think anything about it. It was a silent assumption. Everybody knows that cancer is a genetic disease. I was more or less trying to look.
That's the general thinking for so long, right?
Yeah, me too. What did I mean, I'm a geneticist. I looked at you find all these mutations in tumors. It was very, very complicated to me. I never really could figure what they were looking at or why they had so many mutations, but I didn't care. One word, I didn't care. That was another thing, because I was working on glycolipid biochemistry and epilepsy. And then I mapped many genes in epilepsy, and I learned a lot about epilepsy. And then one of my students here at Boston College said there was a big meeting on ketogenic diets for epileptic seizures in Seattle, Washington, and asked me if she could attend the meeting and write an abstract. And she says, if my abstract is accepted, they will pay my flight from Boston to Seattle. And I told her, no one is interested in ketogenic diets. It's a waste of time. Mainly because when I was at Yale University, I wrote up a small internal grant to of this neurology department, a leading department in the world on epilepsy. And they told me no one cares about ketogenic diets and epilepsy. So several years later, when my student wanted to go to Seattle, I said, if they're going to pay, great, because I'm not going to pay for you to go and learn about ketogenic diets in epilepsy. But anyway, she went to the meeting and we came back, and she was so excited about what she had seen at this meeting. And Jim Abrams, who started the Charlie Foundation, his son Charlie had intractable epileptic seizures and was near death from all these seizures until they went to Johns Hopkins University, where the late John Freeman and Millie, his assistant, gave Charlie a ketogenic diet, and all of a sudden his seizures stopped.
If you want to learn how to implement the ketogenic diet and the Press Pulse protocol into your cancer management, just visit school.com/keto for cancer, the new community by our research analyst Johnny Rockermeyer. Within this new community platform, you can connect with other patients and learn from their experiences. You can also just search through the posts other members have already made. You will have access to our classroom section as well, which provides practical advice and tools for the best results. These sections cover all parts of the Press Pulse protocol that you should implement. Starting with the General Scientific Concepts by Dr. Thomas Seyfried, we will show you how to start the ketogenic diet, including which foods to choose and which to avoid. Additionally, we prepared a list of doctors specialized in metabolic therapy, as well as details about glutamine inhibition and hyperbaric oxygen protocols. So, take a look around at school.com/keto for cancer. If you want to learn how to implement the ketogenic diet and the Press Pulse protocol into your cancer management, just visit school.com/keto for cancer, the new community by our research analyst Johnny Rockermeyer. Within this new community platform, you can connect with other patients and learn from their experiences. You can also just search through the posts other members have already made. You will have access to our classroom section as well, which provides practical advice and tools for the best results. These sections cover all parts of the Press Pulse protocol that you should implement. Starting with the General Scientific Concepts by Dr. Thomas Seyfried, we will show you how to start the ketogenic diet, including which foods to choose and which to avoid. Additionally, we prepared a list of doctors specialized in metabolic therapy, as well as details about glutamine inhibition and hyperbaric oxygen protocols. So, take a look around at school.com/keto for cancer.
So we have all these epileptic models in the lab, and Mariana Tran, who went to the meeting, said, "Why don't we try ketogenic diet on the epileptic mice that we're developing here?" And sure enough, it was unbelievable. So I put the mice on a ketogenic diet, and the seizures became much, much less. So, and then we started to see that these ketogenic diets could inhibit epilepsy regardless of the etiology. Some people inherit epilepsy from a gene predisposition. Some people get a bang on the head. Some people get viral infections. Some people get tumors. So it didn't make any. All I knew was that ketogenic diets were having a suppressive effect on seizure activity, regardless of the etiology, how the seizures came about. So then we did some. Funny thing, we did some calorie restriction. We at the same time in my angiogenesis project, Dr. Mukherjee joined my lab, and she was big on angiogenesis, and we found this ganglioside that would block angiogenesis. So we started to look at calorie restriction in angiogenesis, calorie restriction in epilepsy, stopping both blood vessels and epileptic seizures, and then the two paths overlapped. And then all of a sudden I said, whoa, this calorie restriction works to stop angiogenesis and shrink tumors. Calorie restriction lowers blood sugar, elevates ketones, stops epileptic seizures. So what the hell's going on with all this stuff? And then some drug company gave me a drug that blocked ganglioside synthesis and maybe stop angiogenesis. And we looked at this drug, and yeah, it stopped angiogenesis, shrunk tumors. But what was happening is the drug was preventing the mouse from digesting the food that they ate, and the tumors were shrinking. So it was like an indirect calorie restriction, reducing glucose. You can't get the energy from the food, and therefore the blood vessels were stopping, tumor getting smaller. the same drug also stopped the epileptic seizures because the mouse couldn't eat the food. He lost weight. It's just amazing what happens. But these projects were parallel, but then they started to cross. And then I realized that the reason why the drug was stopping the seizures is because they couldn't get the glucose from the food they were eating. And
I see.
then I realized that had anyone ever seen something like this before? And Otto Warburg said a long time ago that all cancer cells are dependent on glucose to grow rapidly because their oxidative phosphorylation is defective. So I said, well, who the hell is this guy, Otto Warburg? I started around the year 2000. I started to look into this German scientist called Otto Warburg, because I heard the name many times in my education of the Warburg apparatus.
Early in the 1900s, right?
Yeah, well, yeah, Warburg did his work in Germany from 1920 probably all the way up to his death in 1970, and he was working on the cancer problem. And his initial findings said that all cancers are dependent on a fermentation, on a glucose fermentation. So you can't grow tumors without their fermentable fuels. So I said, then I started looking into this more. Excuse me. And then I said, why they have to ferment? And Warburg said their mitochondria are abnormal. They can't get energy from oxidative phosphorylation. And then I said, oh, really? Is that true? So then because of my great background in biochemistry of lipids, I had a student, Mike Kebish, and I said to Michael, first we did a big genetic study on mitochondria. We looked at the mitochondrial genome in all the brain tumors that I had, and we found that not one of these brain tumors had a genetic mutation in mitochondrial DNA. So I ruled that out. Not to say that mutations in mitochondrial DNA can't be involved in cancer. I just said in my 5 independent mouse brain tumor models, there were no pathogenic mutations. So I said, well, clearly the cancer in these mice can't be due to genetic mutations in the mitochondrial DNA. So we decided to look at cardiolipin and lipids in the mitochondria. So Michael isolated and purified the mitochondria, isolated from the 5 different brain tumors. And every one of them had abnormalities in cardiolipin. Cardiolipin is a major lipid in the inner membrane of the mitochondrion, playing a very important role in oxidative phosphorylation. Okay? So all of our models of brain cancer had abnormalities in cardiolipin. So, and we knew from the lipid literature that any abnormalities in cardiolipin would affect indirectly oxidative phosphorylation. So when I saw that, I said, oh, that immediately supports Otto Warburg's theory. Otto Warburg said that problems in the mitochondria were responsible for the tumor cell needing fermentation energy because the mitochondria are defective. So we published a big paper in the Journal of Lipid Research, the #1 prestigious journal in the field of lipids, on the cardiolipin abnormalities in brain tumors, directly supporting Otto Warburg's theory. Unfortunately, the cancer field ignored the study completely. I found out later, I couldn't understand. I thought this was the greatest thing. I said, we have direct biochemical evidence supporting Otto Warburg's central theory that oxidative phosphorylation is abnormal in cancer cells, directly supporting what Otto Warburg said, meaning the cell has to ferment using glucose as a fuel.
Yeah,
So
definitely.
Yeah. So, but the field completely ignored this, and I went to the AACR meetings, the American Association of Cancer Research, to explain to some of the cancer people doing research on cancer that I said I support Warburg's theory. They said Warburg was wrong because cancer cells have normal oxidative phosphorylation, and I said no, they don't. I tested this, and then I looked in the literature, and there's never been a paper showing normal cardiolipin in any cancer. So clearly, cancers cannot use oxidative phosphorylation. But it turns out, remarkably, that almost everybody in the cancer field was studying genes and genomic sequences. And when I said to them that there's an abnormality in cardiolipin, you know what they said? They said, "What is cardiolipin? I never heard of cardiolipin."
They couldn't even recognize cardiolipin as a part of the mitochondria.
Yes, absolutely clueless. So, and then when you try to tell them what cardiolipin is and how it works with the electron transport chain, they have no clue. It was like talking to electric plug sockets. I didn't know what to say. Here I thought it was a great finding that could bring the cancer field back on track, because now I had supported Otto Warburg's theory with a lipid, a lipid that plays an essential role in the efficiency of oxidative phosphorylation because of its location in the inner membrane, and it's abnormal in all these cancer cells. And that means the cancer cell would have to ferment, get energy from non-oxidative source, exactly what Warburg said. So then I ran up again.
There's a problem in the electron transport chain inside the mitochondria. That's the problem.
Yes. And we isolated and purified the mitochondria from these tumor cells. And we sequenced the mitochondrial DNA, and we couldn't find any mutations, but the cardiolipin was always abnormal to some degree or another. So, wow. And then I was like, wow, how come nobody understands this? How is it possible? Well, they don't do lipid biochemistry. They're all focused on sequencing genomes, looking at thousands of mutations, and that's where their brain is. Their brain is not in lipid biochemistry. Have no clue what cardiolipin.
Nuclear DNA, right? Nuclear DNA, even mitochondrial DNA. And the other thing, we have to isolate and purify the mitochondria, and then isolated the DNA from the purified mitochondria, and then we sequenced every single base. We actually corrected the mouse genome on mitochondrial DNA because we sequenced every base, and all of the tumors, not one pathogenic mutation in the mitochondrial DNA of any of the tumors, but every tumor had abnormalities in cardiolipin in the mitochondria. So the mitochondria, every single tumor, no matter the organ involved.
Well, the brain. We were looking at brain at that time, but then since then, other people have looked at cardiolipin in other types of tumors. And in all kinds of tumors, there are some abnormalities in content or distribution of the fatty acids in the cardiolipin molecular species. And we still know that nobody has shown normal cardiolipin in any tumor to this day. But the power of the cancer field was in two areas. Number one, everybody thought it was a genetic disease, so they didn't waste time thinking about cardiolipin. Number two is that many distinguished biochemists who looked at energy said that cancer cells take up oxygen similar to normal cells, and therefore they must have normal oxidative phosphorylation, despite the cardiolipin abnormalities. So even the guys in the cancer field that knew something about Warburg, they all said Warburg was wrong. Cancer cells consume oxygen in somewhat similar levels to normal cells. So I went back and I read all of Warburg's original findings, had some of them translated from German into English, and it was clear that was untrue. All the cancer cells have a level of oxygen consumption that was lower than the oxygen consumption in normal tissue of the same same species, same tissue. But what the cancer field was doing, they were comparing rat and mouse and human tumors all together, bunched together. And you have to compare human normal tissue with human tumor. You have to compare mouse tumor with mouse tissue, rat tumor with rat tissue. You can't jumble them all together and bunch them up. Otherwise, you get no differences between normal and tumor tissue. So you have to clearly specify species difference, tissue difference, and look at normal tissue, tumor tissue within the same species, within the same organ. Then you see that there is a reduction of oxygen consumption in tumor tissue compared to normal tissue. And the guys in the field jumble them all together. and clearly said, Warburg is wrong. Well, you didn't separate species or organs. And I was shocked that members of the scientific profession would overlook those significant problems. And Dean Burke, who was an advocate of Warburg, clearly mentioned the same problems. But for whatever reason, something wrong with the brain of some humans, they completely ignore obvious findings only in light of what they know and what they think the disease actually is. Even when evidence is staring them in the face, they refuse to believe it. So I kept doing this research and finding more and more evidence that mitochondria were abnormal in tumors, and the reason why they needed to ferment, which was energy without oxygen, is because the oxidative phosphorylation was in error. And I know Warburg made some mistakes, thinking that every mole of oxygen consumed equals a certain mole of ATP produced. Every mole of lactic acid produced was producing 1 mole of ATP through lactate. Those calculations are no longer correct. We have now defined what was correct and what was incorrect. But one of our biggest discoveries, which no one knows about, and we've published many papers, they should know about it. There's the third way to get energy. See, Otto Warburg did not know about mitochondrial substrate level phosphorylation. Most of the people in the cancer field have no clue about this. And listen, unless you really, really understand mitochondrial bioenergetics and energy metabolism, you will make mistakes. So what I do in my life, when I'm confused or not clear about something, I seek out those individuals that are world experts on a particular problem. Mm-hmm. So when I learned about ketogenic diets and calorie restriction, I would talk to the late Richard Veitch all the time. Veitch was Hans Krebs's last graduate student, one of the smartest guys, energy metabolism. Same with George Cahill. Cahill and Veitch, the leaders in the field of ketones and energy metabolism. So when I looked at mitochondria...
Starvation, right? George Cahill studied a lot on starvation, human starvation.
Yes, right? Yes. He was the expert on starvation. And Veitch was an expert on ketone metabolism. And then I went to Christos Xenopoulos at Semmelweis University, the world expert on mitochondrial substrate level phosphorylation. So I had this idea that these tumor cells are getting significant energy from a second fermentation pathway. The first one is what Otto Warburg said, and everybody agrees, called the Warburg effect. They get energy from the fermentation, lactic acid fermentation, using glucose as a fuel. Everybody knows that. They're confused about why that happens because they don't understand biochemistry. They don't understand mitochondria abnormal. When mitochondria abnormal, the cell has to ferment, and glycolysis goes up. So, but the big thing, I in my book, I put together a pathway based on the work of Hokachka, interesting guy from Hawaii. He took these animals and held them underwater, these seals and walruses and these kinds of things, a dolphin or porpoise or something. And these are animals that can live underwater for a long period of time. And he strapped them to a board and held them underwater. Can you believe this? And then he took blood from them while they were holding their breath underwater. And he found in the bloodstream lactic acid and succinic acid became very, very high in the bloodstream of these animals. And he said the succinic acid was coming from amino acid fermentation. Wow, amino acid fermentation. So that stuck in my mind.
Amino acid is that?
Yeah. So then I know that cancer cells need glutamine, which is the major amino acid. And everybody in the cancer field, almost everybody, I think everybody, thought the glutamine was being respired, a respiration used for oxidative phosphorylation. Wrong. I predicted that the glutamine was also being fermented because succinic acid, the waste product of the glutaminolysis pathway, was accumulating in the blood just like lactic acid. So lactic acid is the end product of the glycolysis in the cytoplasm. Succinic acid is the end product of glutaminolysis through the mitochondria. So the mitochondria was not using the glutamine for respiration. It was fermenting the glutamine, just like it was glucose. So we use a non-oxidative source. So I wrote this model, and I shared it with Shinopoulos, Christos Shinopoulos, the world expert on.
Let me clarify that again, Professor. The glutamine is being fermented inside the mitochondria as against the glucose being fermented in the cytoplasm, or something like that, to that effect.
Both, both of them. So you get high elevation of lactic acid fermentation from glucose in the cytoplasm, and then you get a succinic acid accumulation from using glutamine through the mitochondria in a non-oxidative way. It's called fermentation. It's called mitochondrial substrate level phosphorylation. Okay, let's go down deeper. I'm sorry if your audience doesn't understand what I'm about to say. There's two kinds of energy that we can get in our cells. Number one, oxidative phosphorylation, where we breathe oxygen, and oxygen serves as a common acceptor of electrons, driving electrons through the electron transport chain, producing large amounts of ATP from the reducing equivalents in the Krebs cycle, citric acid cycle. That's why you and I are breathing right now, okay? Yes. We're breathing because we need energy. You need energy to hear me. I need energy to say something to you and describe this. This is all oxidative phosphorylation used from oxygen. However, we evolved in all organisms that exist on the planet today evolved originally in an environment where there was no oxygen. There was single cells, no oxygen, but they were growing. Where the hell did they get their energy from? Fermentation. Fermentation is energy without oxygen, okay? So all of the cells, most of the cells in our body have ancient pathways that take glucose to pyruvate, and this can happen in the presence or absence of oxygen, okay? Mm-hmm. So what we were looking at then, I read that there's a mechanism inside the mitochondria where we can generate ATP through, let me say again, in the cytoplasm, it's called substrate level phosphorylation.
Phosphorylation. Mm-hmm.
The ATP, a phosphate group, is moved from an organic molecule to ADP to form ATP, substrate level phosphorylation in the cytoplasm, oxidative phosphorylation in the mitochondria. Everybody in the cancer field think there's only two ways to get energy: cytoplasmic substrate level phosphorylation and mitochondrial oxidative phosphorylation. But there's a third. It's called mitochondrial substrate level phosphorylation. This is another form of energy that can come without oxygen in the TCA cycle inside the mitochondria. I found why, when Hokachka held the animals underwater, did succinic acid go so high in the bloodstream? Because the glutamine in the blood, which is so high in humans and animals, was being diverted through that glutaminolysis pathway to make energy in the mitochondria independent of oxygen. So the same organelle can use two types of energy: substrate level phosphorylation or oxidative phosphorylation, whereas the cytoplasm can only use substrate level phosphorylation.
So fermentation.
Fermentation is energy without oxygen. So clearly we were getting ATP, a fermentation mechanism within the mitochondria itself. So the mitochondria have this flexibility. So when oxidative phosphorylation did not work, they were using glutamine, amino acid fermentation. They were using the glutamine as a fermentable fuel, just as the cytoplasm was using glucose as a fermentable fuel. So I put the two together. Now listen to this. As a physician, you'll understand this very clearly. If a person has a heart attack, what you see in the bloodstream is an immediate rise in lactic acid and succinic acid. Both of them go up in the bloodstream. just like the animals that Hokachka was drowning, succinic acid and lactic acid go high in the bloodstream. Why? Because they have to switch to an immediate fermentation metabolism. Now, if the heart attack patient does not get oxygen back into his system, he will be dead. Okay?
Yes.
Now, the cancer, Warburg said, the cancer cell is locked into a fermentation metabolism because the mitochondria are defective. So when we look at all cancers, they're all producing lactic acid and succinic acid. So that tells us right away they're locked into a fermentation metabolism, but the fermentable fuels are glucose and glutamine. Those are the two fuels driving the growth of these tumor cells. So without glucose and glutamine, no tumor cell can survive because they can't use oxygen. No tumor cell can survive for very long in the absence of glucose and glutamine. So I know I said a lot of stuff here, but it represents years of our research in the lab, where I'm working with Chernopoulos and Semmelweis University, and we're measuring fuels and end products as the result. So listen to this. You can, if you and I or anybody drink the cyanide, you know, cyanide is a deadly poison.
It works in the mitochondria, right?
Yeah,
Cyanide.
it shuts down oxidative phosphorylation immediately. You take cyanide, you're going to be dead. The cancer cell doesn't die from cyanide. Okay? Well, that clearly tells you he's not needing oxidative phosphorylation. So cancer cells live without oxygen and live in cyanide. I said, what the hell? They're not using oxidative phosphorylation. I don't care if they're taking in oxygen. They're not using it for ATP synthesis. And now we know that. They're uncoupled and they have other kinds of problems. But where do they get their energy from? So listen to this. We then grew our tumor cells. No glucose, no oxygen, only in glutamine. And guess what we found? We found ATP being produced, continual ATP being produced, and succinic acid being accumulated. Direct evidence that they are using mitochondrial substrate level phosphorylation as the third fuel. Warburg knew nothing about this. He did not know about this. This has confused the entire cancer industry. First of all, they have no clue about most of what I'm talking about. Most of the folks today at the top medical schools are studying gene mutations and chromosomal abnormality. They're not talking about what I'm talking about. And the guys...
They're hooked to it. It seems like they've been hooked to it for a long, long time.
Oh, yeah. Completely brainwashed. Indoctrinated on this gene theory. So what I'm saying, I might as well be talking to a farm animal or something. It's not going to have any impact on the brains of these people. So even though we have now defined what a tumor cell needs to survive and grow, and we have tested these tumor cells, and we can't find any fuel except glucose and glutamine to drive the dysregulated growth of the tumor cell. Okay, we've interrogated all these fuels. We've looked at all these different kinds of things. And I'm telling you, now the other thing, when the mitochondria are defective, they can't switch to fatty acids or ketone bodies. You need a good mitochondria, like Cahill and Veach told me. You can't burn fatty acids unless the mitochondria are efficient and functional. So my big paper just recently published a few months ago. What I did, I went through this scientific literature for years and looked at electron microscopy of human cancer, and every major cancer has defects in the number and structure and function of the mitochondria. Found that out. Then I went back and I said, wow, if they have defects in mitochondria structure and function, they will not be able to burn fatty acids or ketone bodies. Okay? So, and all major cancers, if you look at them under the electron microscopy or in different ways, they all store lipid droplets in the cytoplasm. So you can see all the lipid droplets in the cytoplasm of the tumor cell. Why? Because the mitochondria are defective. The fatty acids accumulate to protect the cell from death, so they accumulate these lipid drops. So every major cancer with defects in number, structure, and function of mitochondria have accumulation of lipid drops in the cytoplasm, a clear biomarker for abnormal mitochondria.
Because they are not being utilized by the cancer cells. That's correct.
Yes, exactly.
Lipids.
Yeah, exactly. You understand that, right? And I put the paper out, Guess what? Almost everybody in the cancer field thinks the fatty acids are there as energy for the tumor cell. How can it be there for energy when the mitochondria are defective and broken? So they're studying all kinds of gene mutations, trying to figure out why the cells are storing these lipid drops. Oh, it must be for the energy of the tumor. They cannot use that. So,
Yeah, I think let me interrupt a little bit, Professor. It's like the lipid droplets are not being utilized by the mitochondria because the mitochondria is defective or damaged. Correct.
yes, exactly.
And
And that,
only the fermentation pathway works in the cancer cells, but these lipid droplets are not being used in the fermentation pathway either. Correct.
that's exactly right. Exactly right. So the lipid drops are a biomarker for abnormal oxidative phosphorylation and the dependency on fermentation. So just looking at and all major cancers looked at under the microscope have these lipid drops. And to see the mitochondria, you have to use electron microscopy to see the mitochondria. And invariably, they're always damaged. There's always some abnormality. Makes perfect sense. Bad mitochondria, lipids have to accumulate because if they don't accumulate, they actually dissolve membranes and will kill the cell. So they're stored in droplets as a protective mechanism. So if you read, and right now, if you and your friends go to the literature and look up lipid drops in cancer, 99% of the papers will say that those lipid drops are used for the energy for the tumor cell. Exactly the opposite of what I'm saying, because they don't believe that mitochondria are defective, even though I show them pictures of this. And this is brainwashing, because cancer must be a genetic disease. So all this stuff you're talking about can't be true, can't be real. So this is the problem. So now we know
it's like almost a dogmatic belief that cancer is a genetic problem then.
Yes.
Yeah.
Yes, exactly. All the stuff I'm talking about doesn't mean anything because it's due to the mutations. So then after I supported Otto Warburg, absolutely supported his central theory, I've cleaned up the mess around his theory with all the misunderstanding and misinterpretation of the data. And now I show a third form of energy that can also clarify that nobody knew about, mitochondrial substrate level phosphorylation, that explains why we have two fermentable fuels, not just one fermentable fuel. And then in the clinical, in patients, and in preclinical models of brain cancer and other cancers, when you lower blood sugar and elevate ketones, the tumors begin to shrink, angiogenesis goes away, and the patients get much healthier, okay? Without chemo and radiation or any of this stuff. So what's going on here? Why is that happening? Because the tumor cell, you're taking away one of the major fuels for the tumor cell. So now, if we put the cancer patient on calorie-restricted ketogenic diets, almost anything to get a low glucose ketone index, putting them in nutritional ketosis, and then on low-carb diets or whatever, and then you come in with parasite medications. Why parasite medications? Because I have found in my research that the parasite and the cancer cell use mitochondrial substrate level phosphorylation to stay alive.
Which parasite medications are you talking about?
It seems like most of them. I have a big article here on parasites. It seems like a lot of parasites use the same pathway. It's very interesting. Parasites use oxidative phosphorylation during their sexual cycles and replication. But once they go and sit inside the tissue, they switch to substrate level phosphorylation. The cancer cell is locked into substrate level phosphorylation. So, but parasite medication by itself has some therapeutic benefit. It's when you combine it with calorie restricted or state of nutritional ketosis. Now you're pulling the plug on both glucose and glutamine. So we have a paper that we've just published in pediatric brain cancer with embendazole, which is a parasite medication. Embendazole has an effect on reducing glycolysis and glutaminolysis, the two fermentable pathways. So Once you understand the biology, all this stuff makes complete sense. You
Is
don't know
this
how to
the mebendazole that is being used by the parasitologists to control the helminths, the anti-helminthic
Yes,
medication?
yeah.
Oh,
Also targets the same energy pathway in the cancer cell. Uh,
oh, I see.
uh, yeah, because I found out why they kill parasites, because parasites use substrate level phosphorylation as an energy source, just like the cancer cell. This stuff is like, what the hell? It's so simple. And everybody says, oh, take parasite medications. Yeah, but if you take parasite medications while under nutritional ketosis, it works a hell of a lot better. Really works better. So what I have told you, and I have the evidence. I'm publishing papers. I mean, what I'm saying is out. You can read the articles. So what I'm saying is that we can manage cancer very, very effectively in the majority, if not all, cancer patients by lowering the fermentable fuels, glucose and glutamine, and transitioning the whole body to ketone bodies and fatty acids. Because the liver burns fatty acids, and the rest of the cells, the brain, can burn ketone bodies. All organs can burn. Cancer cells cannot burn fatty acids and ketone bodies. They store them as lipid droplets. They're not burning them. So you can starve the cancer cells by taking away their fermentable fuels while transitioning the whole body off to a non-fermentable fuel, ketone bodies, which we evolved as a species to burn. So you have to transition the cancer patients from a high carbohydrate diet. You gradually lower the carbohydrates. The body's glucose goes down, ketones go up. And when they get into the zone that we published, 2.0 or below, the ratio of glucose to ketones, you hit them with the parasite medications. And you start gently hitting them with the glutamine glutaminolysis targeting drugs. But you have to be very careful when you do this, because our immune system, when you kill cancer cells, the immune system comes in to pick up the dead cancer cells. The immune cells need the glutamine also. So you must be very careful on how you target the glutamine in the cancer patient. The cancer gut, the health of the microbiome in the gut is glutamine dependent. If you are too aggressive with the glutamine targeting drugs, you could harm the immune system and the gut. So that's why we developed the press pulse therapy for managing cancer. You press glucose down and choke it, because our body can switch to ketones, so you don't need glucose. But the glutamine you must pulse and take off. So you have to be very strategic with the glutamine targeting, because our immune system must come in and pick up the dead cancer cells to correct and heal the cancer microenvironment. All this is completely logical. It's based on the underpinnings of the failure of the cancer cell. They're very inflexible. They only can do fermentation. They can't switch to anything else. So they're very easy to kill if you know how to kill them logically. But what are they doing? They're using immunotherapies, Opdivo and Keytruda, and they're using all these toxic drugs, all based on the somatic mutation theory of cancer, the gene theory of cancer. If the theory is wrong, the outcome will never be optimal. So we continue to treat cancer patients with toxic poisons, radiation, immunotherapies. I'll tell you the reason. The multi-billion-dollar genome project was supposed to provide us with precision medicine, personalized precision medicine. It has been an abysmal failure, this whole thing. That's why we continue to irradiate and poison the cancer patients, because the promise of the genome theory, the gene theory, never materialized. Because cancer is not a genetic disease. It's a mitochondrial metabolic disorder driven by fermentation. So in order to manage cancer, you must target the fermentable fuels, transitioning the body over to non-fermentable fuels, and the cancer cells will die, as the body, as a natural healing machine, will take care of the microenvironment and heal the cancer. This will be the new theory this will be the new metabolic treatment of cancer. Eventually, everybody in all medical centers throughout the world will be treating their patients with metabolic therapy once the brainwashing from the gene theory dissipates. How long will that? I don't know, because the National Cancer Institute continues to tell people cancer is a genetic disease, 100 different types of cancers. That's all incorrect information. I have looked at all those cancers. They're all fermenters. They're not different from each other. Yes, liver is different from brain, different from colon, different because it comes from a different organ, for crying out loud. But if you look at they're all fermenting. Every one of these cancer cells are fermenting. So you can use a singular procedure to eliminate or manage, let's say. I never want to say that metabolic therapy is a cure for cancer. It's an effective, non-toxic management of cancer. There is a chance you could be cured, but there is a chance you may need to live with this disorder for the rest of your life, like our friend Pablo Kelly with the brain glioblastoma, 10-year survivor. The tumor never went away completely, but it grows so slow. Every now and then he gets a resection of the tumor, but he's alive. He's alive.
Pablo is in the UK, right?
Yes. Okay. So here's a situation. Oh, it's just so crazy. So Pablo, N of one guy. He decides no radiation, no chemo, no steroids. And now he's alive 10 years later. And the field says all the patients that they treat with standard of care, 95% of them are dead within 2.5 years. Here's one guy who chooses not to do standard of care, and then he's still alive. Why you don't want to do what Pablo? So the system refuses. The system refuses to stop irradiating the brain. So I talked to the neuro-oncologist. So I said, you need 3 groups. Okay, the group standard of care, 95% of people are dead in 3 years, 2.5, 3 years. 95% are dead. Why don't? Then they say we'll do metabolic therapy with radiation. Okay, we'll do that. They make that. Well, some of these folks are living a little longer, and they're doing better. Not all of them, but we're getting more and more longer term survivors, right? So I said, why don't you do metabolic therapy without brain irradiation? And they said, oh no, cannot do that. And then I wrote many papers showing how brain irradiation frees up massive amounts of glucose and glutamine in the microenvironment of the tumor. In other words, forcing the tumor cells to survive and grow. In other words, contributing to the death of those patients. There has not been one single advance in brain cancer survival for glioblastoma in 100 years. Not one survivor not one major advance in 100 years. And they keep doing the same thing over and over and over again, expecting some different thing.
Yeah. This brain irradiation, as you said, increases the level of glucose in the brain tissue itself. Is this separate from, because we neurologists also give steroid medications to cancer patients. So is this separate from the effect of these steroids?
It's radiation. When your body is irradiated, blood sugar goes up naturally. It's a threat response. Your hormonal glucocorticoids are released. Your body is under threat, so go up. And then when you irradiate the person's brain, there's heating and swelling from all the radiation. So you give high-dose steroids to reduce the swelling from the radiation, which further increases the level of glucose in the bloodstream. And then they wonder why the patients are all dying. I mean,
so you mean to say, ideally, given that scenario, the brain in cases of brain tumors or brain cancers should not be irradiated then?
Oh, absolutely. They should never be irradiated, ever. Right? So you have a tumor in the brain. The first thing you do is powerful metabolic therapy. It will shrink the angiogenesis. It'll shrink down the tumor small. Then the neurosurgeon comes in and cuts out the small tumor that now has a sharp, defined line. The tumor cells come back into the center. When you take away glucose, they come back into the center to get the microenvironment, because there's glucose and glutamine in the microenvironment. So you do parasite medication, calorie-restricted ketogenic diets, shrink the tumor down. The neurosurgeon comes in, takes out the tumor, and you transition the patient back to metabolic therapy. You never bring that person to radiation in the brain.
Are you passionate about the groundbreaking and heroic research of Dr. Thomas Seyfried on metabolic therapy and cancer? So are we. That's why we've created something special for you, in collaboration with Johnny Rockermeyer, a German book publisher and translator. Introducing our collection of meticulously crafted books that distill the essence of Dr. Seyfried's work. Dive into the science and discoveries. These summary books are your gateway to understanding the intricate world of metabolic ketogenic therapy in a clear, concise, and engaging way. Whether you're new to the subject or a seasoned enthusiast, our books offer insights that can change your life. Ready to explore this transformative knowledge? Visit our website at www.cancerasametabolicdisease.com to get your copy. You can buy the ebook there directly and the paper book via the provided links. Here's the best part. A portion of every purchase goes directly to support Dr. Thomas Seyfried's groundbreaking research. That's why the direct ebook purchase is the best option to donate as much as possible. You can see all of the donations Mr. Rockermeyer has already made at www.ketoforcancer.net. That's right, when you buy our books, you're not just investing in your own knowledge, you're also contributing to the future of cancer research. Help us make a difference. Together, we can drive change and save lives.
So I have told these radiation oncologists about this over and over and over again. I published the paper, Lancet Oncology. I published these papers everywhere. Nobody wants to believe it. Nobody. Yet Pablo is, you have how you change the system. So the only way to change the system is to educate the people, because apparently the neuro-oncologists are uneducable. They're, for whatever reason, they cannot be educated. It's there are some people you cannot educate. I'm sorry I have to say these kinds of things, but I don't know what more evidence that I could do other than give myself a brain tumor and not have any radiation and do metabolic therapy to see if I can survive. I mean, is that the kind of evidence that somebody would believe? But I got Pablo and I got other brain cancer patients out there that are living much longer on metabolic therapy if they can avoid radiation. And even if they are radiated, the metabolic therapy can help them live longer, even though they were radiated. But why you do radiation in the first place? And you know what they say? They say, well, I don't believe you because cancer is a genetic disease. Don't you know it's a genetic disease? So what you're talking about is no relevance.
It still goes back to the wrong assumption from the very beginning.
Yes, absolutely.
The genetic cause, supposedly.
I just received today several papers showing how targeting gene mutations in brain cancer can help the cancer patient. I mean, yeah, it's worthless, absolutely worthless. And the federal government is spending all this money on these kinds of studies, toxic chemicals, targeting gene mutations inside a tumor that already has 100,000 mutations. So it's unbelievable how the human brain, once it's indoctrinated, I guess you can say for religion, for political reasons, you cannot convince someone that one religion is better or not better than the other one. Your brain is indoctrinated. The same thing happens to the cancer people. They think cancer is a genetic disease. So what I did, I went out and we found papers that no one likes to talk about. Some cancer cells have no mutations, okay? How you explain dysregulated cell growth if I have a cancer cell with no mutation? Oh, forget about it. Don't talk about it. Then I found out that only some mutations are bad, like driver mutations. These are the bad ones. Passenger mutations, they're just there. They don't do anything. The driver mutation. Within the last 5 years, we do now sequencing in different organs of our body, and we're finding all these driver mutations in normal cells that never become dysregulated in cell growth. How you explain that? And then I did the nuclear transfer experiments. I put that together, where the cancer nucleus loaded with mutations is put into a new cytoplasm, and the results are regulated growth. Whereas if I take the normal nucleus and put it in cancer cytoplasm, I get dysregulated growth. So the driver of the dysregulated growth is in the cytoplasm with the mitochondria, not in the nucleus. Not in the nucleus. Can you believe this? Can you believe what I just said? This is why in the United States, we have 1,700 people a day dying from cancer, 70 people an hour. And most of the people, 50% of the people dying from cancer are dying from the toxic treatments used to manage the cancer. Can you believe this? This is a tragedy of the greatest monumental in the history of medicine. Nothing could be more tragic than the current cancer situation. A massive lack of knowledge, a failure to accept or understand the knowledge that's there so that the status quo is not changed. The status quo must persist. Radiation, poisonous chemicals, and this is what you do, one after another. And I'm telling them, a lot of this is not necessary, and they don't believe me. And I say, here's the evidence. They don't want to look at the evidence. They don't ignore the evidence. So what are you going to do? What's the solution to this problem? Even though you have people living longer, it works. People are living far longer doing metabolic therapy, and they don't want to believe it. So what am I going to do? I just told you, and I publish all this stuff. It's not like I'm keeping it to myself. I'm sharing it with the world, and nobody wants to hear it.
Yeah, and I've heard of others also having the same experience. And you talked about Pablo Kelly. I think you heard probably also of Andrew Scarborough.
Yes.
Yeah,
Yeah.
yeah. Also in
Yeah.
the UK. I think both of them has glioblastoma multiforme, GBM. Is that correct? Yeah, both of them
No,
have GBM.
Scarborough had an anaplastic astrocytoma grade 3.
Oh,
But
I see.
and here's the other thing with Pablo Kelly. Oh, this just kills me. It breaks my heart. He had an IDH1 mutation. Okay, IDH1 mutation. I call it God's gift to the cancer patient. If by chance you get this mutation, the IDH1 mutation by chance acts as a drug that stops glycolysis and glutaminolysis pathways, interferes with the ability of those two pathways to provide energy for the tumor. The mutation produces 2-hydroxyglutarate. It's a product of this mutation. 2-hydroxyglutarate inhibits glycolysis and inhibits glutaminolysis, the two pathways driving the dysregulated growth. Now, here's the crazy. So people with the IDH1 mutation live 2 to 3 times longer than the glioblastoma patient who does not have this mutation, right? So then what they did was they reclassified GBM based on the presence or absence of the IDH1 mutation. So they call it glioblastoma, a high-grade stage 4 IDH1 glioma versus a mutant versus the wild type grows faster, the mutant grows slower, but it's still glioblastoma. So now companies at Johns Hopkins think that mutations are bad. They think the IDH1 mutation is bad. So then they're going to make a drug to stop the accumulation of 2-hydroxyglutarate, the compound that's stopping the growth of the tumor. So they gave a patient the drug, and it killed the patient faster than if they'd done nothing to the patient. But they keep doing the drug. They put drug companies put all this money into these drugs that are taking away God's gift because they have no clue about the metabolism of cancer. So they think all mutations are bad. So they're making drugs to stop 2-hydroxyglutarate, which is giving the patient an opportunity to live longer. Can you believe the insanity of what we're doing out there? This is terrible. So and I've told people this, and I wrote a letter to the company that's making the drug to stop this. I never heard back from them. They ignore me. So they can make money selling these terrible drugs to these cancer patients, inadvertently killing, taking away whatever advantage the patient may have, irradiating the brain, putting that patient in a much greater probability of death than if you didn't do that. Why you do that? Because they need to make money on these patients. This is the key. It's more important to revenue generation than it is for the outcome of the survival of the patient. I hate to say that, but that's the truth.
No drug intervention, no money, then no business.
Yes. Okay. Well, they should know that. Oncologists should say, "Thank you for coming in. Your affliction will keep me, my family, in financial security." So, but you can't say that. And then the other problem is, terrible situation in this country. And not only does many of these drugs cause horrific toxicity, but even if you don't survive, which happens many times, you have to pay an enormous price for these failed drugs, and that causes financial toxicity. So a lot of poor people who earn money paycheck to paycheck they get hit with a bill for $10,000 or $15,000 for a drug that didn't work, and they have no way to pay for this. And then their marriage forms apart. The patients go into bankruptcy. They lose their house. This is immoral. This is immorality at its highest level anywhere. Okay, this is an immoral system doing this to another human being. Not only are you poisoning and killing them, but then you're charging them and breaking apart their finances as the result of this. Can you believe this? Can you believe what's going on here?
That holds a lot of parallelism with this thing about reversing metabolic syndrome and insulin resistance also, because there are so many drugs there that are meant to target diabetes, hypertension, and all the complications of insulin resistance without them really acknowledging the fact that the root cause can always be stopped so that this will not progress and the medications will eventually be stopped. I think that's the same case with cancer as a metabolic disease then.
Yeah, well, we have Guy Tannenbaum, who is on the web. He had stage 4 prostate cancer metastasized to the bones, but he was obese. He had type 2 diabetes, hypertension, and high blood pressure. So the doctor said he's going to be dead in about a year. So he did water-only fasting, low carbohydrate, water-only fasting. His cancer went away. His diabetes went away. He lost 35 pounds. Hypertension, all the things went away, all linked to the same chronic problem. So obesity has now replaced smoking as a major risk factor for cancer. We have more obese people on the planet today than we have starving people on the planet today. Can you believe this?
Well, why do you think obesity is a major risk factor in cancer?
Because it creates...
What's the connection?
It's linked to systemic inflammation. So if you look at C-reactive protein, which is a marker for inflammation, many people have elevated C-reactive protein, type 2 diabetes, coronary artery. They have all these chronic diseases. Besides, they might have cancer. And you do markers for inflammation in the bloodstream, and you see elevated markers of inflammation. So how does inflammation cause cancer? In other words, you can't get cancer if mitochondria are healthy. Chronic inflammation and the cytokines were linked to inflammation, NF-kappa B, and all these damaged oxidative phosphorylation chronically in different cells of certain organs, leading to a dependency on fermentation and dysregulated cell growth. So I can link back all of the origins of cancer. Viruses damage oxidative phosphorylation, radiation, intermittent hypoxia, systemic inflammation, age, viruses, oncogenic papillomaviruses, hepatitis C viruses. All of these things cause cancer by chronic disruption of oxidative phosphorylation, coupled to compensatory fermentation, followed by dysregulated cell growth. The mitochondrion controls the differentiated quiescent state of the cell. When that organelle becomes corrupted, it loses control. Cell falls back on ancient pathways of fermentation that existed before oxygen came into the atmosphere, and all the cells at that time were dysregulated in their growth because there was no mitochondria to control the growth. Everything is completely explainable on the understanding biology, biochemistry, and evolutionary biology. So the obesity epidemic today is evolution in action. Our ancestors that existed during the Paleolithic period survived on very low carbohydrate diets. They had very little sugar availability, carbohydrate in the environment. We had to kill and eat animals. We ate whatever plants we could find that were edible, a very little high carbohydrate diet, right? And we evolved over tens of thousands of years to exist in an extremely low carbohydrate environment. Then, within a period of 100 years, we're confronted by massive amounts of readily available, highly processed carbohydrates. And immediately you get an obesity epidemic. And the epidemic now is associated with all these chronic diseases because the foods are readily available, very little exercise. Everybody's sitting in traffic, sitting in front of the computer doing this and that. No exercise. Highly processed carbs that taste delicious, absolutely delicious. We sought these for existence on the planet. Now they're available, and we eat, get fat, get diabetes, get cancer, cardiovascular disease, dementia. All these things are the result of our advance in food technology and science. So evolution of food technology and science has happened far, far faster than the evolution of our physiology. We're still locked in a Paleolithic existence in a new environment of highly processed carbohydrates, and the results is the obesity epidemic. So people want to know evolution. They don't want to understand evolution. Look at the obesity epidemic. Evolution in action, completely explainable on concepts of physiological biochemistry.
So that explains then some quarters that say maintaining oneself on a Paleolithic way of eating would be of optimal benefit if that's the case.
Yeah, I said you can't. It's hard to get cancer and diabetes if your mitochondria remain healthy. You know, it's. I mean, that's the way we evolved. We're an efficient machine, an energy machine inside of our body, largely controlled by mitochondrial structure and function. So if you're pounding this poor organelle with high carbohydrates, creating systemic inflammation and all of these other lack of exercise, that organelle becomes corrupted. And and then it switches to fermentation, leading to dysregulated cell growth or insulin insensitivity or, cardiovascular disease due to inflammation. All of these things are inflammatory linked to abnormal diet and lifestyle. So yes, but yet we like it. And we like the tasty foods that have come from our ingenuity and technology. But the problem is, in the old days, most people were locavores. Locavores are people that eat regionally grown foods and vegetables.
Are you passionate about the groundbreaking and heroic research of Dr. Thomas Seyfried on metabolic therapy and cancer? So are we. That's why we've created something special for you, in collaboration with Johnny Rockermeyer, a German book publisher and translator. Introducing our collection of meticulously crafted books that distill the essence of Dr. Seyfried's work. Dive into the science and discoveries. These summary books are your gateway to understanding the intricate world of metabolic ketogenic therapy in a clear, concise, and engaging way. Whether you're new to the subject or a seasoned enthusiast, our books offer insights that can change your life. Ready to explore this transformative knowledge? Visit our website at www.cancerasametabolicdisease.com to get your copy. You can buy the ebook there directly and the paper book via the provided links. Here's the best part. A portion of every purchase goes directly to support Dr. Thomas Seyfried's groundbreaking research. That's why the direct ebook purchase is the best option to donate as much as possible. You can see all of the donations Mr. Rockermeyer has already made at www.ketoforcancer.net. That's right, when you buy our books, you're not just investing in your own knowledge, you're also contributing to the future of cancer research. Help us make a difference. Together, we can drive change and save.
They would have a garden, a natural garden fertilized by manure and these kinds of things, and they would eat healthy natural foods because there was no global distribution of food around the planet. People were mostly locavores. They would eat their local foods, and they would be healthier, and they would have a lot of exercise, and they would have much less cancer, diabetes. They were not obese. Okay, only in the last 50, 75 years do you have the explosion of all of these cancers and dementia. Another one, dementia. That's another link to mitochondrial dysfunction in the brain, imbalances of energy metabolism. All of this is perfectly explainable, but we would prefer as a species to take a pill in a cup of water. Okay, take this pill, your cancer is going to go away. Take this pill, your dementia is going to go away. Take this pill, your diabetes is going to go away. So, these new drugs you give, everybody would rather take the pill, Ozempic or something like this, get rid of the, right? It's easy. I don't want to do exercise and diets and all this stuff. So I don't know what the long-term consequences will be for that, but we'll have to wait and see. If it helps somebody, great, But anyway, I focus my attention on cancer. And I understand cancer because I have done the research myself. I have read many hundreds and thousands of papers on cancer. I put it all together. I have the necessary background and the knowledge to interpret data. That's what I was trained to do as a geneticist and a biochemist, interpret data. So I can do this, and I'm putting these links together, but I feel like I must be crazy because no one else can seem to understand what I'm saying.
A number of veterinary medicine friends of mine are saying that, pets, domesticated pets, seems to be having more incidence of cancer than their counterpart that are, living in the wild. What can you say about that?
Yeah, it's
Is
true.
that also the same
Oh,
as how
absolutely.
the human condition is in terms
Yeah,
of
so
cancer?
we found that all domestic dogs are derived from the wolf through our selective breeding. We selected as a species the traits in these dogs that served our purpose, whether it was to hunt down and help an animal or a guard dog or a retriever or whatever. They were selectively bred, but they all came from the wolf. Okay, wild wolves, cancer is very rare in wild wolves. We found out the number one killer of wolves is injuries, infections and injuries, not diabetes or cancer. So why all the domestic dogs are affected with diabetes and cancer, just like the human? Because the dogs are eating highly processed carbohydrates in a bag, and it's cheap. You just throw the food into the dog, eats it up quick, and they have carbohydrates, vegetables. They get all kinds of things a wolf would never eat. Wolves are basically carnivore. They eat chickens and rats and mice, but they don't eat corn, and they don't eat spinach. They don't eat these kinds of things. So they're putting cornmeal and carbohydrates in the dog foods, and the dogs will eat anything. You know that dogs will eat almost anything. And you put a little smell of meat in there, little fragrance of the meat. Oh, they'll eat it all down, and they get fat, and they get diabetes, and they get cancer, just like humans. No difference. That's because we're late.
Modern modern humans are like domesticated animals.
Yeah, no different. Yeah. The guy goes to the oncologist to take care of his colon cancer, and the dog goes to the veterinarian to take care of the. And then it's very interesting because I had a student who was a vet, and they don't use a lot of the toxic chemicals that are given to us for cancer management on the dog, like Red Devil, this doxorubicin that they give to many cancer patients. Makes your urine red, your eyeballs are red. That's why they call it Red Devil. So they can't do that to the dog because it's called animal cruelty. The dog has vomiting and diarrhea and all this kind of terrible stuff from this drug. So they can't use some of the toxic drugs we give to humans. We can't use it to the dog because it's animal cruelty to treat the dog with the same chemicals that we treat ourselves with. I mean, the whole thing is crazy. It makes no sense. And then we published a big paper on the mast cell tumor on the dog's face. I got that published in Frontiers. You should look at that. The dog had a big ass tumor on his face, big on the lip, right under the lip. The woman fed the dog some vegetables and some fish, and the tumor got bigger. Then she read our papers, calorie restricted the dog, raw chicken, fish oil, and raw egg, cut down really. And within 3 months, the damn tumor went away. The dog died from old age, from heart disease, with heart failure from old age, not from the return of the cancer. The dog was very, very healthy after it went on metabolic therapy. Just happened to die from old age.
Professor, let's backtrack a bit to the genetics part because there are certain cancers that have been, let's say, established to have links to genetics, like for example, breast cancer, BRCA1, BRCA2, and like let's say Li-Fraumeni syndrome, which is I think a problem with the TP53 protein
P50,
or gene.
yeah,
Yeah,
P53.
P53. Yeah. What's the connection between these established gene mutations and the metabolic theory of
Yeah.
cancer as you're
Okay.
proposing?
We call them secondary risk factors, not primary risk factors. The primary factor for the origin of cancer is insufficient oxidative phosphorylation coupled to compensatory fermentation. So how does BRCA1, P53, retinoblastoma, a couple of these other mutations? We went through all the so-called inherited germline risk factors, and we found that every one of those genes, in some way or another, disrupted oxidative phosphorylation, leading to a secondary fermentation. So it's the gene mutation itself that codes for a protein that the gene product itself can disturb oxidative phosphorylation. like p53 encodes cytochrome C oxidase 2 in the electron transport chain. Now, the interesting question, so clearly, and everybody that develops a tumor from p53 mutation or BRCA1 or retinoblastoma, they're all fermenters. The cells are all fermenting in these germline-induced cancers. But we looked into it and said, how do we know that those germline mutations are not primary risk factors rather than secondary risk factors? And the answer is no inherent germline mutation has ever been found to be 100% penetrant, which means that everyone who has the mutation demonstrates the neoplastic dysregulated cell growth. That would be, let me give you an example. Huntington's disease is due to a mutation in the gene Huntingtin. Everyone who inherits that mutation will develop Huntington's disease. So the primary cause of the neurodegeneration is the mutation Huntingtin causing abnormal Huntingtin, the protein, leading to neurodegeneration. That's 100% penetrant. That's the primary cause. In cancer, we have never found 100% penetrance between the mutation and the phenotype. So that means the cancer mutations are secondary risk factors and will cause cancer only if the product of the gene disrupts oxidative phosphorylation, leading to a compensatory substrate level phosphorylation. So what is the primary cause of cancer? And that is chronic damage to oxidative phosphorylation coupled to a compensatory substrate level phosphorylation occurring in the cytoplasm and in the mitochondria. That is the origin of cancer, absolutely, and it's been seen all over. The gene mutations are downstream effects. So where do all these gene mutations come from that we see in the nucleus? They come from the reactive oxygen.
Question.
They come from the mitochondria. So when the mitochondria are dysfunctional, they produce ROS, reactive oxygen species, the hydroxide ion, the anion reactive oxygen species. Hundreds of papers are in the scientific literature showing that ROS, reactive oxygen species, are carcinogenic and mutagenic. In other words, they cause the mutations. Where are they coming from? They're coming from the mitochondria. Why are the mitochondria producing all these ROS? Because their mitochondria are defective. The oxygen is not being used for ATP synthesis. It's being used for ROS production, causing the damage in the nucleus. So we spent $100 billion trying to find the mutations in the nucleus, all downstream effects of ROS from the mitochondria. And their effects, they're not causes. So we then designed all these drugs to try to target the mutations in the nucleus and are killing more people than helping people. So it's such a tragedy on every level of understanding, on every level of our knowledge. But I talk to you, I talk to all these other people. I have no idea why nobody understands what I'm talking about. I feel alone. I don't know what to say. And yet all what I talked about is in the literature. It's scientifically established. It's not like I'm pulling this out of my butt. I mean, this is scientifically established truths and facts that no one seems to be able to understand when you put it all together because they're locked into thinking cancer is a genetic disease. And you're 100% correct. When you say germline mutation, oh, it must be a genetic disease. They're secondary risk factors. They're not primary risk factors. They're doing the same thing as a chemical carcinogen, a virus, or chronic inflammation. They're doing the same kinds of things to a particular organ. So some woman may have breast cancer. She had BRCA1 that happened to damage mitochondria. Another woman may have breast cancer. She had an occluded milk duct leading to intermittent hypoxia, causing cancer. Another one may have a chemical. It all depends. You look at it under the microscope, the pathologists say, oh, you got stage 4 cancer. And then we know that how you get metastatic cancer, because the epithelial cells that start are not metastatic. Immune cells come in to heal the wound, fuse with this. Macrophages can move through our body to heal wounds and kill bacteria. They fuse with the stem cell, and now they become metastatic, dysregulated mitochondria, dysregulated macrophages, immune cell. Dysregulated immune cell biology is the origin of metastatic cancer. What do they need to grow? They need glucose and glutamine. So if I take away glucose and glutamine, will I kill metastatic cancer? Yes, absolutely.
So that thing about metastasis, does that explain in terms of cancer as a metabolic dysfunction, does that explain the difference between a benign tumor and a malignant tumor?
Absolutely. So the benign tumors do not have the characteristics of immune cells. The benign tumor has no markers of immune cells like macrophages or dendritic cells. When you look at benign tumors, they're pretty clean. When you look at them away from the tissue itself, because in the tissue you have immune cells that come in, and you have to separate the normal immune cells from the tumor cells. So when you do that, the tumor cells have, the benign tumors have no markers of the macrophage. The metastatic cells, loaded with macrophage markers. So that means there was a fusion hybridization or something where the stem cell fused. Don't forget, macrophages, part of the immune cell, they're highly fusogenic cells. They like to fuse with themselves and other cells to facilitate wound healing. So what happens is they fuse with the non-metastatic stem cells and become these hybrid cells, now having a genetic capability of moving around the body in a cell that biology, that cell can normally do that. Now it becomes a rogue. Why is it rogue? Because the mitochondria in that cell, the mitochondria are defective, leading to dysregulated cell growth and a rogue macrophage that now spreads all over your body. I outlined all this. So then I said, what did they eat? They eat glutamine and glucose. They can't live without it. So boom, out goes. So you can spend $100 billion on trying to figure out what kind of a biology it is. All I know is they can't live without the glucose and glutamine. So why don't we kill all these tumor cells, enhance the health and vitality of the patient, and then go back and figure out what was going on? Why do we have to allow 1,700 people a day die in these clinics needlessly when you guys are not targeting what you're supposed to be targeting to heal the patient? You know, the mechanisms haven't been completely clear. They're clear enough to know how to get rid of the damn cancer. Why don't we go back and figure out all the details, the minutia, after we help the patient survive? What's wrong with that? Why is that so why is that so radical?
Yeah, going to that thing you're talking about, to a great extent, many of these things that we are talking about right now is really controversial to a lot of people. So in your case, for the past years that you have been doing your best to bring this out to the public, are you experiencing any kind of pushback from any quarter, any industry, as a consequence to all these efforts of yours?
Well, I think the push, I don't see pushback on me. When I talk to my colleagues who don't believe anything I'm saying, they just say the field says it's a genetic disease. That's all I can say. Okay, so they just go along mindlessly with what we call confirmation bias. So they don't read I said, why don't you read the literature and see if I'm wrong? Oh, they don't do that. They just reside on what everybody else says. So that's one thing. I read these papers, I see the connections, and then I think I'm crazy. Then I give the papers to my students, and I say, please read this to make sure that I misinterpret the results. So they say, no, Professor Seyfried, these are your idea is correct. And so I say, well, then it's not me, then I'm not crazy. So I have my students as a filter to make sure I'm not crazy, because I read these things. I say, I interpret it this way. Do you see the connect? Oh, yeah, it's very clear. Well, how come the field doesn't see that? So the cancer patients listen to what I'm saying. It's a tragedy, of another monumental tragedy. They hear what I'm saying, get all excited. Metabolic therapy, I want metabolic therapy. Run to the Dana-Farber, the MD Anderson, all these top cancer things. And they go in and talk to their oncologists, and their oncologists say, never heard of it? Must be wrong. If it were right, I would have heard about it in medical school. And they said, glucose has nothing to do with cancer. That's a myth. And the poor cancer patient standing there hearing what I just said, and then go and listen to the oncologist say completely opposite, that if it were important, I would have heard about it. No clinical trials on this, therefore it can't be right. All kinds of things not related to the science. Ask them, did you read the science? Did you understand? No, I didn't do that. Well, how in the hell are you making statements like that to the cancer patients when you haven't even read the science or even understand it? That's the tragedy. That's the tragedy. And then the poor cancer patient emails you back. My oncologist just threw me out of the office because he said all the stuff that I'm saying is a myth and not supported. Well, why don't you read the damn literature? They don't read the literature. They don't understand the biology. And a lot of times it's not their fault. They were never trained in medical school. They were told cancer is a genetic disease because the professors and the MD, everybody in there told them it was a genetic disease. Oh, it must be right. And therefore, the chemical treatments that I'm using are based on the fact that we have to stop DNA replication. We got to poison and stop the DNA replication. They can't replicate DNA without energy, man. Don't you understand? They need energy to replicate DNA. Where are they getting their energy from? Fermentation. Take away the fermentable fuels and help the patient. Oh, no, I never heard of that.
This really brings out a lot of curiosity in me, because all these papers that you published out there, it's available for the entire public to see and for all the other researchers to go through and investigate and read for themselves, and perhaps even duplicate if needed. My question is, has anybody from for example, the American Cancer Society, or perhaps any of the societies of the oncologists, like the American Society of I think Clinical Oncology, for example, has any one of these societies invited you over to present your research into their meetings and conferences, so that most of them would be able to understand this and apply it into their practice?
No.
My big question is why? Why?
Because it's a radical. One at the Henry Ford Cancer Center invited me because of a particular person who had a specific interest, and I told all the radiation oncologists there that their therapies were depriving the patients of longer term survival. And how do you think that went over? Not well. And though it would be one thing for me to tell them that their treatments are reducing the overall survival of their cancer patients, giving them all the evidence to support that. Why do you think I was not attacked? I was not attacked. It wasn't somebody didn't stand up and say, let me show you all the evidence to say why you're wrong. They just sat there, stone-faced, looking at me. Top guys in the field, sitting stone-faced, looking at me. Why is that? If I were wrong, they would have peeled me up and down, because they know they can't attack I'm looking at the evidence, the scientific evidence. They do not have the scientific evidence that I have, nor do they want to look at it. That's the other problem. Why this is not any different than what Copernicus and why Galileo supported Copernicus's heliocentric theory. He had the telescope that absolutely supported what Copernicus was saying, and the Catholic Church refused to even look into the telescope to support what Galileo. So they arrested him for his life. They burned Giordano Bruno at the stake because you're threatening power structures. This is what you're doing. You're threatening a power structure. And when you threaten power structures, they don't want to know anything about it. They ignore it. So it's just and they'll say, oh, Seyfried is too radical. No, I'm not radical. The scientific data are radical to what you think they are. It's the data that are there, and you choose not to look in the telescope, and you choose not to look at the evidence. That's that. So it's just non-scientific. There's something other than science. It's the way the human brain perceives things. Who's right? Very few people look at critically, but it's slowly changing. People, why? Because people want to live.
Yeah, or it is very, let's say, disruptive to a lot of big business that thrives on cancer therapy, I suppose.
Yeah. Well, okay, then cancer patients should know that. The cancer patient's job is to maintain a profitable industry. Thank you very much for your disease. I'm so happy that you have this cancer. It maintains a giant profitable industry. So, but I'm not saying that we have to have a hybrid system. This is a disruptive technology only because it's based on hard science. But it doesn't mean that we can't use radiation and chemo under certain circumstances to enhance the therapeutic benefit of metabolic therapy. There has to be some coming together of standards of care blended now with our better understanding of the science of the disorder, and let the field develop so that new industries, profitable, can replace old industries that were profitable. So we don't want to create too much of a radical disturbance, but a blended change based on our understanding of what the nature of the disease is. So that has to come. It will come easier if we recognize the science, accept the science, and adjust the system to the new reality of what the nature of this disease is. Thereby, everybody benefits. The patients live longer and healthier, and the industries adjust their profit margins in a different direction. But I don't think most people would mind paying a significant price for something that doesn't harm them and gives them the chance to live longer. I think they'll be happy to pay for something like this. But right now, we're paying for toxic poisons that take away, in many folks, their longevity, or if they do survive, their body is so compromised by poisons that they don't live a comfortable life after surviving toxic poisons and treatments as currently given by the standard of care. Let's change this, but not get rid of it, blend it in so it becomes used correctly and more knowledgeably than is currently being done right now. This is what the hope is. The bottom line is how long we can keep terminal cancer patients alive with a higher quality of life and an overall survival. And metabolic therapy can absolutely do that. The evidence is being accumulated. Brad and Maggie Jones' movie on the cancer revolution, they're collecting all these stage 4 cancer patients that are surviving on metabolic therapy. And I think a blended system of standards of care together with metabolic therapy will offer at the beginning the best opportunity to move the needle, the survival needle, and eventually new ideas will come as this field moves forward. So I'm absolutely convinced of this. But how long will it take? Patients need to know, oncologists need to know, scientists need to know. I'm sure and my colleagues that work with me daily know and understand this. They're just as frustrated as I am. They see it, they see the data, they collect the data. How come nobody knows about it? We publish these papers in the scientific literature. And, oh, well, that's a fluke. This is not real for the... I mean, this has to stop. But this is consistent with the Copernican revolution, absolutely consistent with what we saw at that time. The only problem today is if you thought the sun or the earth was the center of the solar system, it wasn't linked to 1,700 people a day dying from dying as the result of your misunderstanding. But today we have that tragedy that's linked to the misunderstanding.
So knowing what we now know based on this let's say proven assumptions that cancer is indeed a metabolic disease, would you say that we can safely say that cancer now has a cure? Would you approach it that way?
No, not yet. Not yet. I can't say that. I don't know, because if you have cancer today and you take a metabolic therapy and the tumor seems to go away and you live to be 98 years old and die from old age and you never had a recurrence of the cancer, only then can you say metabolic therapy cures cancer. But suppose you took it today and 15 years later the tumor comes back or something along these lines. Okay, so were you cured? No, you were given a longer progression-free survival, they call it. When does progression-free survival extend into overall survival? We don't know that yet. We have to treat patients carefully. As we speak right now, we have a major treatment protocol under review, comprehensive for glioblastoma. Once that treatment protocol with over 40 authors, 35 authors on this paper, MDs and scientists who've looked at the data, looked at the strategy, we have gone through a very comprehensive analysis of patient responses that could happen or might not, how to adjust all this. It's a comprehensive treatment protocol for glioblastoma that could easily be used for all major cancers. But we use glioblastoma because almost everybody dies that has GBM. So if we can manage GBM and keep people alive 10 10 times longer, 5 or 10 times longer than would be expected. Obviously, we know what the hell we're talking about, and we know what we're doing. So we would then we would modify that for all major cancers. And then you're going to be beginning to see the clinical trials. And we have to certify MDs. We're setting up a certification program in Switzerland so that people who have an MD license to practice medicine can become certified to treat patients with metabolic therapy. So it's happening. It's an educational mission up and down the scale. The physicians need to know what to do and how to do it. And the system needs to know that it's a mitochondrial metabolic disease. And that's why what we're doing will have optimal results, far greater than what's currently being seen with standards of care, which are largely based on an incorrect theory of what the nature of the disease is. So clearly, it's a work in progress. I'm happy to be a spearhead on this, only because I have seen the evidence to support what I am saying over and over and over again, and how much we can improve what I'm saying. This is just the tip of the iceberg. This is just the beginning. And whenever you start a beginning, you can improve it significantly with an understanding. So this is the beginning of a new realm in cancer. Cancer being treated as what it should be. It's a metabolic disorder, and it can be managed much more effectively once people come to understand the science and the outcome of what's happened. So I have no question and no doubt about it. Absolutely no doubt about it.
Let's say divert a bit to diet and lifestyle, Professor. So because it has been an established fact already that a high carbohydrate diet or a diet high in sugar, for instance, has led to a lot of metabolic diseases that are considered as modern diseases nowadays. So would you say that since based on the Warburg effect and the fermentation of glucose as something that propels the growth of these cancer cells, would you say that this causes cancer? Like, for example, the high carbohydrate diet is causing cancer.
It's not. First of all, sugar itself is not a carcinogen. Okay, so glucose is not a carcinogen. Sugar is not a carcinogen. In other words, if I put sugar on a mutagenic assay, you don't get mutations from sugar. High carbohydrate diets consumed in excess, you have high fat carbohydrates together create an inflammatory system. So it's the chronic inflammation in the body produced from a consumption of high carbohydrate, high fat diets. It's not just high fat without carbohydrate. You lose weight on those diets. Okay, if you like ketogenic, but again, we calorie restrict everything. That's why I developed the glucose ketone index calculator here at Boston College, so that anybody eating whatever diet they want can stay within a range of therapeutic ketosis. But yes, if you eat a Snickers bar or a jelly-filled donut, you're going to fly right out of nutritional ketosis real fast. And same with our Paleolithic ancestors. If they were to eat a piece of ripe fruit at a critical point in the season, their nutritional ketosis would be reduced to some degree. Honey is another example of a sweet food that our ancestors would have wanted. But the bees protect honey, and that can be used. And of course, the biggest origin of carbohydrates come from the Neolithic period, where we learned how to grow grains, wheat, corn, or rice, these kinds of things, that had a lot more carbohydrate, giving us a lot more energy. But everything is in balance. Today, we have highly processed carbohydrates with minimal nutritional value in an environment with minimal exercise, in a highly stressed environment. This climate change is freaking people out. Sitting around looking at a cell phone all day long and a computer, getting fat. All of these things together contribute to an unhealthy physiology, thereby leading to cancer, diabetes, hypertension, all the things that you mentioned, so-called chronic diseases. So if we understand what the origin of this is, at least we can do something within our power to mitigate at least some of this. But right now, most people never heard that glucose is a problem. We see little children that are morbidly obese. This is a form of child neglect, but the parents are not trained to know that. So when you have a lack of knowledge, the outcomes can be catastrophic throughout the world. And what I'm saying is that you just need to understand the science. That's why I spent a lot of time here at Boston College emphasizing scientific literacy. It's important for your health and vitality to be scientifically literate. And a lot of times we don't have that, either in the media. And we're constantly confronted by contradictory evidence. Oh, he can eat that, it's healthy. No, he shouldn't eat that, it's unhealthy. The pyramid is upside down. No, the pyramid's right side up. What is the poor guy on the street supposed to do? He says, to hell with it. I'm just going to do what I do all the time. I'm going to get fat and I'm going to put myself at risk. So you just need to know what to do and how to do it. You need to be scientifically literate. And cancer puts the fear of God into you. So believe me, cancer is one way to address all these chronic diseases. If I can mitigate that, reduce that. So that's where it stands.
So in that case, if you are saying that the ketogenic diet has a big role there, whether it is, especially if it's calorie restricted, would you say that a carnivore diet, pure meat diet, which more or less would put the patient in strict nutritional ketosis, would that be more or less as effective?
You know, everybody is different metabolically to some degree. There are different weights, different cultures, different sexes. Everybody is a unique metabolic person. But what we have to do is we have to say what works for that person. Does a carnivore diet work for him, a vegetarian diet, a Mediterranean diet, a pescatarian diet? Whatever can bring that glucose ketone index into 2.0 or below is the zone we're trying to hit. So again, that depends on the individual, how healthy they are, how we move from one state to the next. And that's your job. The job of the physician is to know what is the best metabolic strategy to bring that person into nutritional ketosis. Would it be a carnivore for this guy, a vegetarian for that guy, a Mediterranean for that guy? We don't know. But then you monitor and see how the glucose ketone index is going. Then once you're in that zone, you say, yes, my patient is now in nutritional ketosis. Then you give them the parasite medications, high IV vitamin C, hyperbaric oxygen, all of these kinds of things that will super enhance the health and vitality of the normal cells of the body and targeting the tumor cell. But each person may be different. That's why the knowledge of the physician in working with patients, they make a decision with that patient to see what would work best for that person. That's why one approach may not be applicable to all patients. So this makes the practice of medicine very, very exciting, because now you're working with every individual as an individual, but knowing what commonalities you might see by changing these parameters. And some person may be tweaked in this direction or the other direction, depending upon what the physician sees and the overall health, how that person feels about the treatment. You know, oh, I can't eat fish, oh, I can't eat vegetables, or I can't eat meat. Well, then we'll match the diet for your particular situation, and we'll see how well we can get you down into those zones. So again, it's going to be a work in progress, but it's going to be exciting, because for the first time, you're going to see dramatic results, and you'll see, I have a strategy that I've seen work better on this kind of a guy than that kind of a guy. So you write it down, and then they share it with others, because it's going to be a whole new world of seeing how well people respond based upon new things rather than the same thing over and over again. So I think it's going to be a real opening to medicine that we have not yet experienced.
Yeah, still within the realm of diet, Professor, because many have said that meat, eating meat promotes cancer or increases the incidence of cancer. If I remember it right, I think even the Harvard Th. Chan School of Public Health even mentioned something like that, to that effect. But the thing is, when you eat mostly meat, you mostly keep yourself on a ketogenic state. So how does that reconcile with these researchers that say that eating meat promotes cancer?
Well, then all
What's
of
your
our
take on that?
Native Americans should have been riddled with cancer. Eskimos should have been riddled with cancer. Paleolithic period should have been devastated. They ate meat all the time. Eskimos eating blubber, walruses and fish and meat and all this kind of stuff. Why these people never had very low cancer? It makes no sense. When you look at the studies done from the Chan school, they're not eating just meat. There was a bun in there. There was other things that were in there, that it wasn't just meat. And how much, don't forget, we evolved. Everything has to go back to our evolution. Who were we? Who were we as a species? Where did we come from? Okay? We evolved just like all other species in an environment that was harsh, cold, arid. We had to survive on anything that would crawl, walk, or fly on this planet. We were carnivores, omnivores. We had to arrive through that. And when you live in that traditional diet and lifestyle, diabetes, cancer, high blood pressure, all that stuff is much less common. So they were meat eaters. Why are those folks didn't have all kinds of cancers that we have today? It has to do with highly processed foods. These meats are raised in hormonal and chemical environments. We're eating too much of it, along with the baked potatoes and other things that are all engineered to be sweet and tasty. So if you go back, let me show you somebody who's eaten in the wild. He eats elk, right? He eats kangaroos. He eats these kinds of things, natural things, right? Bison, buffaloes, and this kind of stuff that are organic. Those guys don't have cancer. Very little, very rare to get cancer in those environments. Okay, sit them here in New York City and Boston, coming out eating big burgers with baked potatoes smothered in sour creams and salts and all this kind of stuff, with a big dessert at the end. You know, you're putting yourself at risk for all these kinds of things. But I would say highly processed carbs are lack of exercise and highly processed carbohydrates probably are linked more closely to chronic diseases than is eating meat by itself. Show me a guy who goes to a restaurant and says, you know, I'll have a filet mignon that big, a little bit that, and that's the only thing I'm going to eat now for 3 days. Believe me, his cancer risk is going to be extremely low. This is the way our ancestors were. We didn't have big meals 3 times a day. Come home, sit down, have a big meal, eat a big breakfast. I mean, we're constantly on the move. So we have to say the majority of our existence on the planet was hunter-gatherer, trying to track down, kill something, and eat it. We're only since the Neolithic period, the last couple thousand years. And then only within the last 50, we had the highly processed world distribution of highly processed carbohydrates. Foods I have Twinkies here. You see these Twinkies right here? Okay. I bought them 10 years ago. Okay. I bought these Twinkies 10 years ago. Look at it. They look like they're brand new, right? They look like they're just fresh. They're a little stale, but they look perfect. I had a mouse that broke into the box and ate one of these things, a 10-year-old Twinkie. And I'm saying, wow, you know, he didn't die, but he probably didn't get healthy eating it either. The thing of it is that's high processed carbohydrates right there. You eat enough of these every day, and you're going to get diabetes, dementia, high blood pressure, and cancer if you eat enough of these kinds of things. So, yeah, I think they're an enjoyable treat. You know, once every year, eat a Twinkie. Once every 6 months, have a Twinkie. It's not going to kill you. But if you put them every day along with highly processed foods and all these other things, don't be surprised if you get cancer and dementia and all these other kinds of things. So, yeah, highly processed carbohydrates, lack of exercise, emotional stress, you put it all together, you got diabetes, you got all these chronic diseases, cancer being one of them. Not a mystery.
You mentioned earlier, Professor, about this press pulse strategy that you are bringing out for cancer patients. So my question is, can you expand a little bit on that? How is that done? And if ever some people, or especially those inflicted with cancer right now, would like to undergo the press pulse strategy, what will they do? What are their options? Can they go to their oncologist and ask for something like that with their oncologist?
No, he thinks cancer is a genetic disease, so he's not going to help you. And they're very. And the other thing that's a problem with the oncologists is they're locked into the standard of care. Even if they want to do metabolic therapy, they can't do it. They're going to lose their. They could lose their license to practice medicine. So if you come to them and say, "I want to press pulse metabolic therapy," they never heard of it. Number one, can't do it. Number two, even if they did hear about it. So how do you address? You have to retrain the oncology field. So pressed pulse is just lowering blood sugar and pulsing glutamine. Okay, so what we do, what not me, but my physician colleagues, they take the patient, they do a complete blood work and see how close or far off metabolic homeostasis they are, put them on a zero carb diet for about 10 or 14 days, get them back into metabolic homeostasis, and then give them anti-parasite medications. The best medication, which is the glutamine targeting drug DON, is not available. They said, it was too toxic. So I went back and looked at the human data from the early days by McGill, and the toxicity was not nearly as bad as Red Devil, doxorubicin, or some of these other drugs they give you. So why they threw it up? Well, they said it didn't work. It only worked on some people. Well, you never targeted the glucose at the same time you targeted the glutamine. You got to target the two fuels together to get optimal patient outcome after the person is in nutritional ketosis. So all of the tools for cancer management effectively are already developed. We don't need anything new. We just have to know how to use the tools better that we already have. And that pressed pulse therapeutic strategy allows you to use better the tools that are already available in the tool chest. We have them. We just got to have people that know, in this case, we use that tool. In this case, we use this tool. We don't just do what we're doing because the outcome is abysmal today. So pressed pulse therapeutic strategy is based on evolutionary biology, an understanding of the biochemistry and biology of the very tumor cells that exist. And that's based on the facts of our understanding. That's what pressed pulse, and it will become eventually the standard of care. I just don't know how long it's going to take.
So I have an audience who was asking, because she knew that I was going to talk to you. She would like to ask you this. I think she's based in the US. How could I get my oncologist to work with you and your protocol rather than going with the conventional route of treatment for cancer? Is there a way for that presently?
Yeah. I don't know. This is the problem. This is the singular biggest problem. How are you going to change the system? I feel terrible. I feel good because I understand the science. I enjoy talking about it to other people. I've looked at it. But I get terribly frustrated when the patient gets all excited and goes to the oncologist and gets slapped down saying, everything that I'm saying is a myth. There's no evidence to support what I'm saying. And the poor patient gets so upset and doesn't know what to do. That's why I give a kit of information out to the cancer patients, at least gives them some knowledge. And then maybe they can talk to somebody. I tell you, some of the oncologists, I mean, they didn't go into this field to hurt people. They went into the field to help people. But the system is training them to use therapies that are not effective for the majority of patients and harms so many people through the toxicity. I think it has to come from several sources to change the system and allow that person who answers the question to get an appropriate treatment from a knowledgeable oncologist. Because she'll ask the oncologist what I'm saying, and he never heard of anything that I'm talking about. How is that possible? So he needs to be educated, and he needs to know what to do. And the oncologists themselves must help change the system. Are they capable of changing who is the system? Who is the system controlling what these oncologists and how they treat their cancer patients? Who writes the standard of care? The standard of care should never have been written in granite. It has to be modified as new information comes along, but yet it is not modifiable. I think maintenance of the status quo prevents these oncologists from making the necessary adaptations so they can blend standard of care. Why are they not allowed to blend standards of care with metabolic therapy? Why do they refuse to blend standard of care with metabolic therapy? Why is the system so rigid that allows no flexibility in what these people are doing? So all of these are I can't address that. All I can do is give the scientific evidence for what I'm saying and why it should be done because of the nature of what the nature of the disease is. But then the system must adapt and recognize for the best interests of the millions of people that are suffering from cancer, they need to make the change. There's no need for why we have all of these people brutalized by a system that does not want to change for a variety of reasons. How do you do that? That poor person goes to the oncologist. Says, I want Seyfried's metabolic therapy. Oh, no, it doesn't exist. Never heard of it. Well, how is she supposed to get treated then if the very person helping her has no clue about what he's doing? Not because of his inadequacies, because of his training in the system telling him what must be done. So this has to be viewed. Patients have to put pressure on their oncologist, put pressure on the system. How you change the National Cancer Institute of the United States of America should understand what the hell I'm saying. Yet they don't or refuse to. Now, how do you explain that? How do you explain the head of the National Cancer Institute putting on their website that cancer is a genetic disease, despite the massive evidence to say that it's not? How do they feel comfortable sleeping at night? Well, they've never heard of what I'm saying. Why? Well, they never read the articles. And most people say, what do you think? What do you think? Oh, no, he's wrong. Oh, okay, he must be wrong. Well, they're not reading the literature. They'll listen to some other fool, another dolt. You know, the problem here is it's systemic all over. How is it going to change? I don't know. But people basically want to live. That's the motivation. The motivation is that if you don't adapt, you're going to be dead and die miserably. So you're going to have you're going to have to change.
Corollary to that, what message do you think should be imparted from your end to doctors the world over, like, for example, clinicians like me who are at the forefront of managing cancer patients, and especially the oncologists, the cancer specialists?
Here's what I would suggest. Look at our press pulse therapy. Look at it carefully. Look at what I'm saying and the articles that I'm saying. Cancer is a mitochondrial metabolic disease driven by glucose and glutamine and transition the body over to nutritional ketosis. I have outlined what needs to be done to do this. Why don't a few clinicians on their own, and you don't have to be an oncologist to do this, by the way. I mean, you're doing blood work. You're looking at blood work, for crying out loud. A patient's blood work. Oh, look he has low vitamin D, his triglycerides too high, all this kind of stuff. You can, as a physician, say, okay, I want to put this patient on have them do a low carbohydrate diet, measure your glucose ketone index, and when you hit 2.0 below, we're now going to give you parasite medications at dosages that are known to be non-toxic to patients. So we already have all the evidence. See how the patient responds. Do CAT scan, MRI on the tumor. See, oh, it was there, now it's gone. Oh, the lump was big, now it's smaller. Wow, let's tweak it this way, tweak it that. Based on the experience of the physician, knowing how patients respond to certain procedures, he can be recording the data on these patients themselves. And I'm writing the case reports. Look at Pablo Kelly's case report that I wrote. 5 years of him measuring glucose ketone index. Who would do that? The patients play a very important role in their success. So they have to realize, as a cancer patient, you are no longer a bystander. You are now an active participant in your health care. So they have to work with the physician as a team. The physician is telling them, measure your glucose ketone index. Then the patient says, oh, I'm not eating any carbohydrates, but my GKI is still 20 above. Well, then stop eating everything. Go on a water-only fasting. And then they'll say, well, I don't know if I can do that. Well, then you obviously don't want to live. You have choices. You want to live or you don't want to live. You get your GKI and the metabolic and the parasite medications thrown in on top of that, and then you start to see the change. And if you had Don and some of these other glutamine targeting drugs, you'd see a tremendously improved. And we're still working out the details. I'm not saying we have the solution to all. All we know is all cancers, or the majority of all major cancers, need fermentation. How do we best target the two fuels driving the fermentation while putting the patient in nutritional ketosis? And that the physician and the patient work together as a team to do that. And now we have monitors on the arm, so you can get information to your cell phone. The cell phone can go right to the physician's laptop, and he can be looking on a real-time basis. All of a sudden, he's got a beautiful GKI, and it springs up to 50. What happened? Oh, I didn't know that banana would have that kind of an effect on my blood glucose. Don't eat the banana. So, I mean, right now we have the technology to make this a really happening thing at a reasonable cost, not something that's going to break the bank. So it's coming. We have everything available. We just need to know how to use it correctly.
Yeah, precisely. That patient of mine, the one I talked to you about, the non-small cell lung cancer with multiple brain metastasis, does survive for quite a longer period of time than what's expected of him. And if he really did that strict ketogenic diet, then yeah, I think it would be worth doing a case report.
Well, you got to, I would say, get the glutamine targeting drug, the parasite medications. You can use
embendazole.
Yeah, ivermectin, embendazole, fenbendazole, all of those. Once your patient is in nutritional ketosis, then you can launch these various parasite medications as cocktails at the same time. And if you have hyperbaric oxygen, they can sit in the chamber when they're in nutritional ketosis. That'll put oxidative stress more on the tumor cell than on the normal cell. So all of these things can be add-ons. And the goal is, how long can I keep my patient alive with a higher quality of life? You know, that's the singular goal. Yeah. And I have guys, I see people email me several years later that say, hey, listen, I'm still alive. I thought this guy would have been dead, and he's still alive. So even Pablo Kelly is still alive. But I'm seeing many, many people alive, and they're happy. They're not suffering. So I consider that great success. You'll see it too. Once you start looking at what we're doing and modify it for your own patient population, and have the patients record their own glucose ketone data, so they know what they're doing and why they're doing it. Now you have a compliant person. You know, some patients are non-compliant. There's always going to be a guy who doesn't want to do anything. Well, forget about them. That's his misfortune. We've got to focus on the people who really want to help themselves and work with you. I am not in the business of trying to take somebody who doesn't want to be compliant. I say, hell with them. I don't care about you. I'm only interested in the guys that want to know about this and want to stay alive for their family and other members. So, see, I'm not like a physician. I don't have to be a physician. I'm not a physician. I don't tolerate guys that don't want to know and this stuff. I throw them the hell out. I want to know who's going to be good, and because I need to get those data. And once the world gets the data, then it's going to be more of a standard of care. So we need to have good compliant people. And when Pablo Kelly became non-compliant after his first surgical debulking, he started to eat sweets and high carbohydrate diets again, and we saw the tumor return. It came back.
Really?
Oh, yes. And then it put the fear of God back into him, and he went back, and the tumor. Oh yes, it's unbelievable. I mean, it's going to be you're going to see it with your own patients. It's going to be unbelievable. So, and then when they're non-compliant, the tumor will tell them that they are non-compliant. You don't have to tell them; the tumor will tell them.
I'll surely be getting in touch with you over and over and over again regarding these things.
Yeah, well, once we get the protocol written, then you'll have it in hand, and then you can use it as a tool in your own clinic, and your buddies, your friends can all tap into the same thing, and you'll start to see these dramatic changes. Unbelievable. You'll come to see it. It's just a matter of time. I just got to get it out, published.
Yeah, that's very wonderful indeed.
So we really had a very, very informative and quite extensive conversation out there, Professor. So really very grateful for that. Thank you very much for all those that you have shared here in this podcast.
Should anyone listening to this podcast session right now would like to follow your work or perhaps support all your efforts in cancer research? Where may they follow your go-to?
Yeah, well, thank you very much for that. All of my support to do what I am doing comes from private foundations and philanthropy. There are people who want to be part of this new paradigm change, and support me through either giving money to Travis Christofferson's Foundation for Cancer Metabolic Therapy, which is a foundation that supports our research, Travis Christofferson's Foundation for Cancer Metabolic Approaches or Therapy.
I'll put that in the show notes below this
And
podcast.
also the Boston College, my Boston College biology page at the university. If you put my biology page, there's a donation. It's called Boston College Cancer Research, and people can make a donation to my research directly through the university, or through Travis Christofferson's foundation. Or some people say, I have my own foundation and I want to support you. They can support me from their own foundation through the university. I am not capable of taking any money personally from anybody. I have to go through either private foundation or the university. So that supports me through the institution. And then I hire people and I test more drugs and procedures in my preclinical system to make sure it works and it's not toxic. And then my clinician friends, my physician friends working in the clinics apply that to their patients and let me know how it's working. So my research sets the platform preclinically so that the folks, my colleagues in the clinic, can apply it to their patients. And then they feed back information to me. I readjust my preclinical system, improve it, and I send it back to them. So it's a back and forth from the preclinical to the clinical, from the clinical back to the preclinical. And I use that information, and that's how we constantly improve the system to make it better for all people.
Do you think that's also applicable to people who would like to ask for your assistance outside the United States, or is it just available there in the US?
Yeah, I get hundreds and hundreds of emails from people around the world. No question about it.
Huh.
Yes, from the Philippines. Yes, from China, Australia, New Zealand, you name it. Everywhere in the world, people are emailing me. They all want to get knowledge, and they want to know what they can do to help either themselves or some loved one in their family. And the stories I get are absolutely horrific. I mean, I sometimes have to step away from what I'm reading, from the torturous activities of people being treated by terrible, terrible things. So, yeah, so any event, yeah, our research is supported by private foundations and philanthropy. And as long as we continue to get the funds, the more we're going to improve the system and make the change faster. So the more money that I get to do my research and publish the scientific articles and peer-reviewed publications, the faster the paradigm will change.
I do hope we can all work towards that common goal of really controlling and eradicating, hopefully, the scourge of cancer in the world today, or even just significantly diminishing the rate of death from cancer today, which I think, based on the fact that cancer is indeed a metabolic disease, that is indeed achievable.
Yeah, absolutely. Yeah. I agree with you.
So with that, I thank you very much, Professor Seyfried, for everything that you have explained here in this podcast, and I hope everybody listening in have indeed learned a lot out of our conversation today. So again, thank you very much.
Well, thank you very much thank you very much, because it's your podcast that gets the word out as well. So your podcast allows more and more people to hear the information. So you are just as important in a way as I am. So thank you very much.